Hello, we're here in San Francisco on the last day of the conference on retroviruses and opportunistic infections, otherwise known as CROI, here in San Francisco. My name is Jeff Taylor, and today I'm going to be interviewing Dr. Per Bengtsson. Dr. Bengtsson is the Chief Medical Officer and Senior Vice President of Development for Bionor Immuno, a Norwegian company that has some very provocative immune-based therapy treatments, and we'd like to talk with him about those today. So thank you for coming, Dr. Bengtsson.
Thank you very much. It's a pleasure.
So we've been hearing a lot about the VAC4X study that has been going on for over a year now. Would you like to describe what VAC4X is, how it works, and what you hope to get out of the study?
Yeah, VAC4X is a cocktail vaccine of small peptides, and the peptides are modified, so they are not native peptides, and the modifications have been done in order to make them more immunogenic. And there are also other advantages. We think that we can circumvent problems of energy and T-cell exhaustion by the modifications. So we've seen pretty interesting results in previous phase 2 study, and that's why we follow up with this larger one.
Okay, so tell me about the larger study that you've been conducting.
It's a study we filed with the FDA in 2007, and it's conducted in 5 countries, in the United States, Spain, Italy, and the UK and Germany. Altogether we recruited 137 patients, and the study is still ongoing. All patients have been immunized, and the last patient will complete the study in the summer this year, and we will then have the results available during October.
That's exciting. Now, is this a phase 2 or phase 3 study?
This is a phase 2 study, yeah.
So it's not yet a registrational trial. You'll have to wait for the results.
No, it's not. It's not. No.
Terrific. So what do you hope to do once you get the results of this one? What are your plans for this product?
We we hope that the study, of course, will confirm the positive results of the study we did in Norway in 40 subjects, and then we would be able then to show an effect on CD4 counts and also some viral load reduction. We hope for that, and the study is designed as a treatment interruption study, but we do not pursue that as a regulatory trial. We want to pursue before the FDA. So we're now focusing on combination therapy, where we give the vaccine together with the antiretroviral therapy. But we are also interested to see whether we can give it to treatment-naive patients. So these are the more interesting cohorts currently. Okay,
so what future studies do you have planned for the VAC4X?
Well, the nearest study in time will be a study in patients that have a low CD4 count. This is patients that do not regain CD4 counts after initiating antiretroviral therapy. So about 20% of patients do not respond adequately to antiretroviral therapy.
But they are virologically suppressed.
Yes, they are virologically suppressed, but they don't repair their immune system. And we have seen some results from the previous study in Norway that we can increase the CD4 counts and get a nice response in those patients. We saw CD4 increases in the total number of patients, 40 patients, but it was very modest. But if we looked into patients that had a CD4 count, baseline count of 500 and below, then the increase was more pronounced, well, so that's why we follow up now to see whether we can demonstrate this in a better designed study.
Well, that's exciting, because I know at the last session they just finished with, Dr. Joe Aron was talking about the problems of longevity for people who don't have full immune reconstitution, and this would be an exciting therapy to help repair that, and perhaps.
Yeah, we really hope that this can show some hope for these patients, because it will be an add-on therapy on antiretroviral therapy, and where we can increase the CD4 counts, we hope, with maybe 100 CD4 cells average for those patients below 300 baseline.
Right. Well, they definitely put them out of the woods in terms of opportunistic infections and things
for those borderline, it would be very important.
Terrific. Well, tell me more about the company itself, Bionor Immuno. You're based in Norway. What other products do you focus on? And
Bionor Immuno was founded, was a diagnostic company from the beginning, and the founder, Birger Sørensen, he had some quite innovative ideas around how to make broader specificity of diagnostic kits, and he succeeded in doing that, and he also got his kit endorsed by the WHO, and it was actually selling in parts of the world, but bigger companies with bigger marketing muscles outcompeted that. But the kit is still around. It's a very good kit. But he developed the science around that, and eventually turned into therapeutics. So Bionor Immuno was spun off from Bionor, which was the parent company. So and since then, and this idea to modified peptides has become a platform technology, and by the modification we can then amplify the immunogenicity, and we also will present a different antigen to the native counterparts. I mean, so the immune system will see the new antigens as new, and not the old native ones, and thereby we can then attract naive T cells and get clones expanded that are not exhausted.
So do you have other products besides the VAC4X that you're
yes,
looking at?
Yes, we do have that. VAC4X is targeting cell-mediated immunity, but we are also doing a B-cell vaccine to address another part of the HIV problem, and that's the immune activation, where we address a certain part of GP120 protein, and we've found actually very interesting results here, that we've been able to correlate with sera from patients that are long-term non-progressors, and we're developing that, and we're actually now having I would say very provocative nice results with long-term non-progressor sera that correlates to the peptide that we have designed. And this is a vaccine that we will take into phase 1 next year. And if it works out like we believe it will, then we will have actually 2 types of problems that we can address.
So you envision that perhaps these 2 products could be used together to
yeah,
address,
so you get a synergistic effect and hopefully a better response.
Well, certainly inflammation has been one of the key words coming out of this conference and in previous conferences in terms of not only HIV infection, but all the metabolic syndrome that they're seeing. And that's one of the huge, has a huge impact on quality of life for people with HIV and as well as longevity. So if you can address this problem, it would be very exciting indeed.
Yeah, I think my interpretation of the state of the art now is that the pathology is more or less depending very much on the immune activation, and if you can address that, I mean, you could really achieve something, I think, for patients and the clinical problems. But of course, you need to combine different therapeutic tools.
Right, absolutely.
Yeah.
Well, and this might also have applications beyond HIV and other autoimmune disorders like
it could have,
arthritis.
yes, could have, but again, I mean, we're targeting now an HIV-1 specific problem, so it will be really interesting. And one interesting aspect is that HIV-2 does not have this part of the GP120 protein, so we're addressing something specific for HIV-1, and it's interesting that HIV-1 is actually the more aggressive type.
Right.
Was causing the immune activation more pronouncedly than the HIV-2. So that's part of the background and theory that we hope then that we could actually address this problem of HIV-1.
Great. Well, thank you very much. We appreciate your time. Do you have anything else you'd like to add about?
No, thank you for this interview. Very interesting. Pleasure.
Great. Thank you.
Thank you.